Okay, can everyone hear and see me? Sounds good. All right, so for those of you that don't know me, I'm Luis Solano. I'm in the Swarup lab. Today we're going to be talking about single nuke ATAC, QC, and analysis. And I know it says workshop. Hope it's not too much of a bait and switch here. It's going to be more of a demo and showing off of some of the functions and some of the plotting that you can do with these tools. So not quite a hands-on bit, but you're welcome to come and reach out to me. We'll talk offline and talk about how to set up the environment, and I'm happy to maybe share a notebook or some of the resources used to kind of generate these. Anyway, jumping into things. So I'd like to start by grounding us in the biology. So a pretty important question we can ask is, what is single nuke ATAC-seq even measuring? So if you have, I guess, if we're starting off with chromosomes and DNA is wrapped and compacted super tightly around our histones and our chromatin, and eventually you end up with sections that are open chromatin, or essentially there's spacing between these nucleosomes to the point where gene expression is possible within these open sections. What is happening in single nuke ATAC-seq is you have transposomes. TN5 is going into these gaps and then literally cutting them out. And at the same time, depending on the technology, this might vary a little bit in the specifics, but tagging on a barcode onto those fragments. And then these fragments are what goes into the sequencer. And then this is going to turn into our raw data. So the next question we can ask here, again, kind of rooting things in the biology, is, okay, Sai is having trouble seeing. Can you both see on Zoom? Okay. Okay, let me know if I can do something so that it's more visible. And like it's visible on the laptops, right? Not on that screen. Okay. All right, I'm gonna keep going, and then yeah, let me know if there's something I can do to fix it. Okay, again, rooting things in the biology here. One of the most important plots that we can get just from very basic QC, and it doesn't matter what tool you're using, whether it be the Python-based tools or the R-based tools, you're going to end up with one of these counts by fragment size distribution plots. And this lines up really nicely with a nucleosome-free region and a mononucleosome peak. This nucleosome-free region, this typically, or rather, the mononucleosome peak typically peaks around that 147 base pair mark. And then that's kind of the distance that your DNA is winding around a single nucleosome. And then that dinucleosome peak is essentially just, you have kind of two of those structures, and so on and so forth. This is one of the more basic QC plots that you get from almost any ATAC-seq analyses. The next plot that is super important for figuring out quality control is going to end up being this TSS enrichment score versus the number of unique fragments. And so every tool has a way of calculating this TSS enrichment, which is essentially, can we find where transcription start sites are in our fragments? And then are there more of these in known and annotated transcription start sites than there are in, say, our background? So typically, you end up seeing something like the following. And of course, all of your tools are going to be pulling from these beautiful data sets, PBMC, BMMC, where you know that the data is high quality, the library preps were solid. You're not really worried about quality when you're working through your, I guess, your tutorials, because they're already giving you something that they know works. So you're going to see these TSSE scores that are well above 20, very well separated groups. And generally in these, you are looking for high quality cells to be in this upper right quadrant. I'll come back to that slide in just a little bit. But if you're more used to tools such as Archer or snATAC, these plots might be a little bit more familiar. Ultimately, it's just coming down to different ways of slicing and dicing your TSS enrichment or your unique fragments. And you can approach QC in these the same way you might approach, say, transcriptomics, where you would take outliers or the tips and the tails of these, cut those off, and then see what the data looks like that way. And then that would be a very basic naive analysis to just kind of get things started. The last metric here is going to be the distribution of the nucleosome signal. This one is from the Muon, I guess, package. And in this one, if you go through their documentation, they pretty much state that the lower quality nucleosome signals are going to be from 2 and onward. So you want that distribution to have lower peaks. Okay, moving on. So often we don't just want to query healthy cells. We're actually often interested in pathological states. And where this becomes a confounding variable is that the biology of the pathology itself can degrade your cell quality. And if that is the case, that can make the analysis process and the QC process specifically quite challenging. So for instance, the Swarup lab, we deal a lot with Alzheimer's disease. So let's take this case study just to have something that we can concretely talk about. Let's assume an AD mouse model and that we particularly care about a gene that is not normally expressed in that. We'll call it gene 1 or gene X. And samples 1 through 4 will be our control mice, and samples 5 through 8 will be an overexpressed gene, again, that we care about. And we can ask the question, what chromatin accessibility differences exist in brain region A versus B in all of our different detected cell types after exposure to that overexpressed gene 1? So as I've kind of been alluding to a little bit in these last couple slides, PBMC, BMMC, these are going to be absolutely beautiful toy data sets that it's hard to get bad data from it. And our confounding variable here is that the pathological AD state is going to be damaging a lot of the cells that we are capturing. And that is going to make QC a little bit trickier. So we have to really think carefully about our QC choices and then what that means for our analysis pipelines. So again, it doesn't matter how skilled you are at the wet lab process of the library preparation. It's not going to do much to increase the quality of the unhealthy cells themselves. And if we are working in a system where we know that these cells are damaged or unhealthy, again, you're going to have that confounding variable to work around. So if we go back to that TSS enrichment versus our unique fragment plot, the important question that we can ask for on a per sample basis is where can we place our thresholding cutoffs such that we capture high quality cells or at least cells that are representative of the sample that we're measuring. And then this slide is just kind of giving you an idea of how many cells, the distribution of those nuclear fragments, and then the distribution of the TSSE enrichment. These are essentially contour maps. They're very similar to the ones that are generated by Snap. The only difference here is that I've added the distributions on top here, so it's a little bit clearer where the threshold would be cutting off those the samples. And then if we were interested to do this in whether it be Archer or snATAC or R-based pipelines, any of these plots can be generated with matplotlib or Seaborn, any number of Python plotting packages. Essentially, we can make our ridge plots, our violin plots, and then base our quality control thresholds based off of these if you're more used to working in this framework. So the proposed framework specifically for SnapATAC2 and for scPrinter is going to be the following. Both of them actually import fragment files directly. And I'm going to come back to this whitelist because essentially we need to generate this whitelist with our QC process within SnapATAC first. So after importing the fragments in SnapATAC, we are going to quantify and QC those fragments, perform dimensionality reduction and integration, do some peak calling, motif enrichment. This one is going to be CPU based. And then the output from this process is going to give you a per sample and data H5AD file. And then at the experiment level, we're going to get an and data set, which is H5ADS. It's a different data structure that essentially can store backed and data files. All that means is that the and data file lives on storage and is directly accessed by the and data set and directly manipulated so that it doesn't have to load the whole and data into memory every time. So you make kind of in place, I guess, accessing and manipulations of those and data objects, and then it gets stored in that and data set. The other output that you'll get from this is a gene activity matrix that's also going to be in your H5AD file, as well as a cell by peak matrix, also in that H5AD file format. Once we've completed our SnapATAC pipeline, we can take the barcodes that came from these from the per sample outputs and essentially say, this is going to be our whitelist of all of the fragments that are that we generated with our initial sequencing assay. We can only subset those to the barcodes that we know are quality controlled from our SnapATAC pipeline. So we do import the fragments, but we are subsetting to the barcodes that we know are good quality or that we know we've already vetted. After that point, scPrinter has you do what they call generate groupings. And if you're familiar with, say, DSeq2, when you're creating your contrast matrices, that's what this process reminds me of. You're really just setting up the comparisons that you want to make, and you're doing it in a way that scPrinter's data structures will understand what you are trying to compare. The next thing you can do with scPrinter is a GPU accelerated chromvar motif enrichment analysis. And this is useful for both transcription factor as well as candidate cis regulatory element analysis. So if you're looking for enhancers, this could be useful. The last two things that happen here are kind of go hand in hand and are really where I think scPrinter shines. It's their transcription factor binding scoring combined with their multiscale footprinting. And then this is applicable for both transcription factors as well as hypothetically candidate cis regulatory elements. A little bit more on this now, but I'll kind of come back, or a little bit more on this later. I'll come back to it. So let's get an idea of what these SnapATAC workflows look like. I'm going to briefly share a couple of their tutorials. So if you go to SnapATAC's, you can access SnapATAC2 through the scVerse. They have all these tutorials. There's a couple things to keep in mind. I'll try and kind of point out what they are as we go, but you start off with just importing the libraries and their fragment files, pre-processing. This kind of explains something very similar to what I did a little bit just a few minutes ago, and shows some of the plots you can generate. It also shows you how to go through some doublet removal, dimensionality reduction, your clustering, and then this imputation process is how you generate your gene activity matrix. And then these are also useful for making your cell type annotation calls. One critical thing here is this close operation. Because and data objects are open in backed mode, if you don't do this dot close, you can accidentally corrupt all the work you've done up until that point. And I can't tell you how many times this has happened to me, but close your files. Anyway, from here, most people will go on to identifying differentially accessible regions. And then this is just the process of once you have your cells annotated, I want to know the difference in the chromatin accessibility from either cluster to cluster, or in this case, cell type to cell type. And then, so yeah, they have it at a cluster level. This can just as easily be at the cell type level. And then this is an excellent sanity check for making sure that the cells that you have called are what you think that they are. And then this is going to end up being the result of your CPU-based motif enrichment analysis. You end up with a nice little heat map here. Admittedly, I have not gone through the regression-based differential testing, but this is also an option should you wish to do so. The next thing I'll point out for SnapATAC, and this is the last one, is going to be their multi-sample pipeline. So this is essentially the specific syntax to use when you have multiple samples. Most of the time, you're not going to just be processing one, at least if you want to be statistically sound about things. So this is just kind of giving you the framework for how to use their particular framework for processing multiple samples at a time. This is kind of the condensed version of that standardized, of the standard pipeline we've already gone through. It does so through that and dataset object that we briefly talked about. It also goes through batch correction, two different kinds, one being harmony, the other being MN correct or MNN correct. For the data sets we've been working through, I've seen slightly better performance off of MNN correct, but if harmony works better for you, definitely use that. And then you go off to your basic clustering and peak calling, and again, they're reminding you to close your objects when you are done using them. Going back to our PowerPoint. So some practical differences I found from my own use cases with SnapATAC2. This reference file paths. If you are working on your own Linux computer, you're not really going to run into this issue, but specifically on HPC, your home directory might run out of space particularly quickly, and it's useful to place your reference files in a lab server or in your personal public server, somewhere where you have a little bit more space to work with. But it is not super obvious how to route SnapATAC2 because it's expecting to find those reference files in your home directory, but it's ideally you would route it elsewhere to where you have enough space for all those reference files as well as all the data that you are working with. The next thing is that the import fragments works significantly faster if you've sorted those fragments first. So if you're like me and you end up having to run this pipeline more times than you can count, you can earn a couple minutes this way just by sorting them preemptively and then importing those sorted fragment files. Archer provides excellent ATAC support, but if you're trying to stay within that Python environment, Seaborn and Matplotlib are great for plotting whatever you are missing from Archer. Muon is great for nucleosome signaling or nucleosome signal, as well as future integration with multiome. The other thing to note here is samples can be very different, and it can be appropriate to apply sample level filtering. The commands given in the tutorial tend to be like they're global. They would apply the same threshold to all of your samples. And it takes a little bit of Python wrangling in order to get to this sample level filtering, but it is possible and I do recommend it. The last thing I'll mention is that for peak calling, if you just use the internal libraries to do this, it will work, but I found that it sometimes hits errors when it gets to weird regions of chromosomes. So if you filter this ahead of time to just standard chromosomes, peak calling should go a lot more smoothly. And I've mentioned this again and again just because I don't know, pain. Close your and dataset object. Don't corrupt it because then you'll need to run it again. So this part, I'm calling this vibe thresholding just because it's somewhat arbitrary where you end up setting the lines. It's where you feel you're using your best discretion to say, I'm capturing enough of the cells that I care about. And I like the red bar when it's about here. We'll call it good. And what you end up doing to, I guess, check yourself on those assumptions is once you've annotated your cell types and made your gene activity score matrix, make a dot plot out of that. You can treat this similar to how you would treat an RNA-seq analysis. And then if these markers are lining up close enough to what you are expecting, it is a good sanity check. So eventually you'll hit this point where you're making the decision, we can continue to clean this up or we can keep moving. For the sake of moving on in the pipeline, let's assume that you are happy with this particular dot plot and this particular single nuke ATAC UMAP. So that next step is going to be peak calling and motif enrichment. This I have taken from your Archer documentation and tutorial sets, and it's basically describing what is happening during the peak calling process. You might have a couple different cell types, and they might have peaks called in different areas within that same sample. And at a sample level, when you end up merging these, this is the merge peak set that you end up with. And the way that these end up being called depends on the specific, where here they call it overlap and merge. This is a specific algorithm that each tool will apply. And you just kind of have to choose the one that makes the most sense for your given data set. I've left mine at the SnapATAC and Mac S3 defaults. But ultimately, this is what ends up deciding what gets called a peak, what doesn't get called a peak, and then merges everything together when it's all done. Once you have those peaks done, one of the powerful things about ATAC is that you can then ask, there are all these motifs for transcription factor binding sites that I already know from previous experiments, from databases, from annotations that I know are important, or that I'm curious about how these transcription factors interact with my particular data set. And you can see where within these called peaks do we have enrichment of our motifs that we know correspond to specific transcription factors. And in this way, we perform transcription factor motif enrichment, and we can use a similar process for enhancer motif enrichment. So that kind of takes us to the tail end of our SnapATAC2 pipeline, and we now have everything ready to go into scPrinter. The last thing I'll note here is that should you want to use scPrinters, I think the slide is a little bit out of order, but basically this was just pointing out that if you wanted to use scPrinter for its GPU accelerated Chrome var, it performs something very similar to the CPU-based motif enrichment, but it does so with GPU acceleration. It's a little bit fancier. Maybe you trust Chrome var a little bit more, but scPrinter gives you a way to do that. So with scPrinter, there's a couple different plots that it generates. Everything from raw signal at the sample level to kind of like where those fragments are piling up, transcription factor binding scores, and then they have these multiscale footprints, which are super cool. And I'm not going to pretend to be an expert on these. I can share my interpretation of these with you, but if anyone knows a little bit more on how to interpret these, definitely feel free to correct me, or feel free to elaborate if you happen to know more about these. But basically, these are, you can think of them as footprints or as kind of shadows that a transcription factor or that a protein might cast upon your fragments as they bind to DNA. So when they are bound to DNA, those areas are no longer accessible, or they have more limited accessibility by your transposomes, and that's gonna make a footprint or a shadow, or it's gonna cause a difference in your background that you can pick up. And based off of that footprint, we can start to make inferences on what protein is bound to that specific DNA sequence. And they call it multiscale because you can ask this question when your DNA viewer window is at the scale of, say, an individual motif, which is just a couple dozen base pairs. You can ask what it is at the level of a promoter, which might be a couple hundred base pairs, or you can ask at the genome level, or rather at like the chromosome level, and each of these is going to give you a slightly different information. But scPrinter allows you to visualize these kind of all in one within the same figure within the same plot, and I do think that's some pretty powerful functionality.
What is literally on those plots? I'm sorry, the axes, the y-axis is cells, correct? And if so, how are they sorted?
Right. So, okay. So the tutorial, unfortunately, I don't know why they didn't label their y-axis. So that's part of my own confusion as to like, and like the way I'm explaining this is, uh, they have in the paper that each row is essentially a scale. And so you can imagine this to be like 500 or 1,000 or so different scales that you're getting measurements at, with smaller scales at the bottom here and then larger scales up at the top. Or I might have that flipped, but the point is that it goes from smaller viewing windows to larger viewing windows. So
Yeah, that's so then I always thought the x-axis was base pairs.
it is. It is.
Okay.
Yes.
Yeah.
The X and Y axis are both base pairs, but one of them is like the base pair of the DNA binding region, and then the other is like the size of the footprint left by the DNA binding protein. It's more of like a I guess a spatial perspective almost.
Is it like
Yeah.
a score that they do?
Kind of. Also, please take all this with a giant grain of salt. Please read the paper. My understanding on these might not be perfect. They are very cool plots, and I do think there's a lot to learn from these, but I don't have a perfect understanding of them. And I can show some of these with a little bit more context with our own data. These are all from the tutorial itself. So a couple helpful notes and practical differences from the tutorial. This reference file path, this is similar to SnapATAC in that scPrinter expects its reference files to be in a specific place, and it is helpful to place them somewhere where you have plenty of space to work with. So just kind of keep that in mind and route appropriately. The barcode whitelist, I've kind of mentioned this a little bit. This whitelist just comes from the quality controlled and data objects that you generated during the SnapATAC pipeline. One thing or several things that are not obvious from the tutorial that you kind of have to, somewhere between reading the paper and then just messing around with the provided functions and the code. It's not super obvious how to identify nucleosomes, though we know that nucleosomes would leave footprints because they are proteins that bind DNA. Similarly, specific transcription factors are specific sizes and will bind a certain known amount of DNA. And you should get transcription factor to transcription factor resolution with this tool. And they show that in the paper, but the tutorials don't provide the code for how to do that. So this is those two. And then the last one here, candidate cis regulatory element analysis. So essentially, how then do you link enhancers and potentially even the transcription factors that are binding to enhancer regions how do you link that to this multiscale analysis? I do believe that there's a way. The paper definitely includes a way, but the specific code to do so isn't inherently clear from the tutorial. Although it is, I'm betting it's somewhere in the functions or even a function to come, and it's that they had ready for the paper, but not necessarily for their documentation. One closing note here before I go through to a couple examples is also sensitive here that this is an AND data that uses backed AND data, so make sure to close it. Otherwise, you'll be really sad when you try and access it later and it says it's corrupted and you have to rebuild it. While it's not super clear how to specifically identify nucleosomes or how to specifically identify a known transcription factor footprint, there's a couple things that might help here. One of which is ENCODE provides something known as screen for candidate cis regulatory element identification. And essentially, we can use this in combination with some of the provided scripts from scPrinter. And then this will let us get at enhancers that are already known, already documented by, and have been like essentially orthogonally verified by ENCODE, or at least orthogonally proposed by ENCODE. So this has kind of been the approach that I've been taking to my own enhancer identification, or proposed enhancer identification. Moving on to grouping. So this is what I had mentioned was similar to how in DSeq you might have to make a contrast matrix in order to tell it what comparisons are important. So it creates a dictionary of annotations and metadata, and essentially you want to map those conditions to your known barcodes, and then again, there is specific functions within scPrinter that will generate that grouping, but you do need to kind of format dictionaries in such a way that it's ready to accept those. I would highly recommend when you go through the tutorial on these, inspect the data structures of their toy data sets, and that gives you a really good idea of what these functions are expecting. The source code is also available on GitHub, so you can definitely look through those. And that'll also give you clues as to what this is expecting a little bit more explicitly. And then at the end of that grouping, you end up with, at least for our purposes, we wanted to know a cell type specific kind of condition conscious comparison. So for any given cell type, we wanted to know for that condition, was there an accessibility, was there a transcription factor difference between the two conditions in like essentially our brain regions. So yeah, grouping is important, and I would recommend spending your time here and making sure it's correct. So what does this actually end up looking like, and maybe how do we do a little bit of interpretation on these? So here we have 3 different examples of what this multiscale footprinting might end up looking like. In our very top row, we are going to have our raw fragment at the sample level, raw fragment data. And so you kind of have your fragment pileups for each one of, for I guess each one of these cases. And we can zoom in a little bit. Okay, so for these, the y-axis are going to be your ATAC signal, and then your x-axis is going to be the specific DNA region that you are interested in. What does the fragment pileup look like in that particular region at a sample specific level? The next thing you can do is, after we had calculated transcription factor binding scores, we can ask, is there a difference between our control and our treatment groups? And then in this particular case, we have increased transcription factor binding scores in our treatment group, at least for this portion of the DNA region under query, and then contrast that to, in this case, where our control was higher and we had a decrease in our transcription factor binding score in our treatment group. The next thing that I'd like to draw your attention to is that for that footprinting here, you'll notice that there's 2 different colors going on. For our original, zoom out. For the way that for the way scPrinter does this, they have it essentially it's just one condition, so it just has one color. But because we care about differential, call it multiscale footprinting, what I did is essentially overlap the signal from the control onto the signal of the treatment group, and then it is only colored in the case where some threshold has been passed, and otherwise that area is grayed out. So it might still have a transcription, it might still have multiscale footprinting at that area, but let's bring into a specific area so it makes a little bit more sense. So like something like here, let's see if there's a better example. So the more gray areas are going to be, here we go. So like something like here, there's clearly a shared DNA protein binding interaction that is occurring here. It is very similar between control and treatment at this scale, but then there's a differential in, I guess, as you, go down in scale. So with a larger scale, the control has more of, that footprinting score. And then as the scale gets smaller, your treatment group goes up. And then this is kind of like maybe a transition zone where there's not too clear of a difference between our control and our treatment group. And then our last row here is earlier when I was saying that each of these are rows, and this could be like 500 or say 1,000 different multi-scale samplings. You can consider this to be the specific row of, if you were to think of this as a heat map, this is the specific row of that heat map around the 7 base pair region. So around this region right here, you're taking out this sliver of the footprint and then asking what is the difference, or where do we see more signal from our control versus our treatment group. And then it's the same paradigm where the more neutral, wherever there is no real difference, it's going to be this white, and then red is higher in treatment, and then blue is higher in control. And you can do this at several scales here, 7, 22, 52, 82. And these could be indicative of different DNA protein binding events. And then the reason I highlighted these 3 specific cases is, again, you have a case where the treatment group is essentially, it has higher footprinting scores, a case where the control is a little bit higher, and then a case where it doesn't seem like there's much of a difference between the two at all, despite several, like we have a lot of reads in this region, but it doesn't seem like there's much of a difference between them. Although here, there does seem to be some differences at the multiscale footprinting level, but the transcription factor binding score level, these are pretty much right on top of each other. And then, of course, we could inspect each of these a little bit more. Like at each scale, we can start to make hypotheses about what DNA protein binding interactions might matter. One of the last couple of things I wanted to talk about was cell-specific average genome-wide transcription factor binding scores. You might be a little bit more familiar with these from other tools, but essentially these kind of take for a given transcription factor and for a given cell type, what is the average binding score that you see at that at the known motifs where that transcription factor binds, and is there a difference between your control and treatment groups when you, ask this particular question. A couple future directions here. Earlier, I said I felt like it was vibe thresholding because all you're doing is saying, I like the line when it's right here. I would prefer this be a little bit more either automated or objective. So I am interested in pursuing potentially an automated or a learned QC thresholding method. I do want to hone in on enhancer and TF motif window centering. So earlier when I was showing these, it's possible to center, to show a line essentially where your enriched motifs are within these bound regions, or the DNA protein bound regions. And I do think that would add another powerful element to this analysis. Yeah, the last parts of this are just how do you know what specific proteins are mapped at what levels? I suspect we're going to have to go like in depth at each one of these levels and be like, where would a nucleosome leave a footprint? And then kind of work backwards from there and work backwards from the specific transcription factors that you know that you have motifs for. Another future direction here is just integration with true single nuke multiome. And then this is going to be for when you have single nuke RNA-seq analysis to go with this. You can obviously, those are going to be complementing technologies. Lastly, I am definitely still learning myself. So if you have any suggestions in terms of how to make this better, I'm more than open to suggestions, and I'm happy to take any questions if there are any.
I have a basic question. So we have already existing tool like Arca and snATAC. What is the advantage of this tool?
Yeah, no, solid question. The question for those on Zoom is why this framework over existing things like Archer or snATAC, more well-established tools and pipelines. For me, the biggest draw is going to be this integration with, is going to be the integration with multiome, specifically because Muon allows you to, integrate single nuke RNA-seq as well as single nuke ATAC. For me, there's a strong argument to be had for moving into the Python framework, and SnapATAC is, in my opinion, one of the better ways of doing that. So nothing wrong with Archer or snATAC. Those are really powerful technologies. It's just that if later on I know I need to move into multiome, I think this is the better tool for the job.
Question. So integration with SN multiome or generally, like where are those sites of integration? Is it actually during the integration process you want to be considering both of those?
Yeah, no,
or where else is there?
yeah, good question. I have not truly started the multiome integration parts yet, so I don't have a great way of answering right now. But I do know that because Muon has, the capacity to integrate both your ATAC and your RNA-seq signal, and SnapATAC gives you a way to QC that data in Python. I am, making the bet that it will be easier to move into Muon, given that it is also based off of AND data and kind of those data structures.
So, and I also kind of missed like where does Muon, because you mentioned the N data set object. Is that specific to SnapATAC2, or is that where Muon is coming in?
My understanding is that Muon takes advantage of the AND data and the AND data set data structures, and as does scPrinter, as does SnapATAC. So I'm assuming that those, and they're all on SC versus the other thing.
Yeah.
So like I'm assuming they will play nicely with each other. It will be somewhat easy to move data from one modality into another.
Yeah.
I have a question
Yeah.
about your QC thresholding for the data you use. Are you using some defaults that is just recommended by SnapATAC tutorial? Because like when we're doing single nuke QC, we know we don't want mitochondrial, full brain, whatnot. So I'm curious, there are some like well-established QC metrics for
The tutorial. Yeah, the tutorials have better, literature and documentation on that. But what I was aiming for, because we know that our cells are not healthy, so that's like whatever those guidelines are, like they're basically going to tell you to take the cells in the upper right quadrant, but our upper right quadrant is kind of empty. So, are we discarding 90% of our cells, or is it the case that we need to work with the cells that we have and then QC on metrics that aren't strictly just TSSE enrichment? What I ended up doing is essentially I'm trying to set these lines such that I capture the greatest density of the I guess the cells that are on that right-hand side. So a greater number of unique nuclear fragments, and then trying to set some lower bound cutoff where we're not taking too much of the actual garbage that I'm assuming is going to be down in this bottom right.
But even knowing what fragment size we're choosing to go with, I think that's a hard one. Right?
Yes.
This depends on like your tissue or the question that you're trying to study.
Yeah. Right now, just by eye and just on vibes, to put it that way, I've basically been taking like where there starts to be like kind of this curve in the second peak in that top distribution, and that will tend to line up with like the lower end of the nuclear fragments to take. And then the bottom of this, there's like another valley here to look out for. And then that's what I've been using for like the bottom of the QC, if you will. I don't know if that's the best approach, but that's the approach I've been using. Seems to be working okay. And then just a question in the chat about accessing the video. We'll see if we can get you that video, Malika.
Yeah. That's very cool. So if you have like a bimodal distribution, like would you consider like the modes? So you could consider like, I guess, everything that you see in the data when it comes to QC. Like it's just to me interesting how you have like two different distributions, especially in the control 5 except 80. So I don't know if that helps you.
Yeah, no, just kind of like brainstorming off of that a little bit. So the clean data sets tend to look like this, and they just tell you to like cut here and like here, and then like they're obviously telling you to get this set of cells. But now you have me curious, like what would happen if you isolated this, brought that through the pipeline? What does that actually look like? Presumably they're not good cells, but I don't actually know what it does look like. So yeah.
Yeah, like lower quality cells.
I mean, could you automate that selection, just pick the steepest part of the curve on the distribution?
Working on it, yeah.
And then more, again, like a basic question. Does a higher binding score means like you're more confident that the binding is happening at that level, or does it mean like there's just more interaction at that location?
That's my understanding. I guess I'd have to look at the specific source code of the function that calculates that to see how it's calculated. But my understanding is that it's working off the assumption that if the transcription factor is bound, you're going to get like this flanked pattern of where TN5 can actually get in there to make the fragments. So it will calculate a score based off of like that. It's looking for that pattern essentially, but I don't know exactly how it's calculated.
Are there theory? I haven't read the scPrinter. I'm looking at it now. Are there theories about what sorts of transcription factors are exerting their effect and which scales? You mentioned how you'd have to go through each. Like, why does it go from 0 to 100? And what resolution would you look at the scales?
Yeah, no, that's a really cool question. I think I don't have a great answer for the transcription factor question. But from what I remember reading in the paper about the nucleosomes was if you know that nucleosomes are going to bind approximately 147 base pairs of DNA for every like wound around, that will leave a particular footprint. So that might give you a good clue as to how to find the nucleosomes in those footprints. Presumably a transcription factor of, I guess, that binds at a known motif should have like a certain amount of DNA base pairs that it like displaces as it binds, and then that would also leave its own characteristic footprint. I think that's the intuition for how to think about it, but I don't know what that would look like, say, for like a tiny transcription factor versus like a massive transcription factor. Yeah. Cool. Well, I guess with that being said, happy to talk with anyone offline further about this. For now, I'll cut the recording and then, yeah, thank you all so much for coming.